All right. Hey, everybody. Thanks, Tim, for the introduction. And thanks to everyone here for listening. My name is Andrew Hansi, and I'm a fish biologist with the USGS Great Lakes Science Center stationed at the Hammond Bay Biological Station. And I'm here today to provide an update on the SWIM project, which stands for Spawning Whitefish and Invasive Mussels. So I'm going to begin by highlighting the primary motivation for this project. So as we all know, and as has been highlighted earlier today, the distribution and abundance of Dreissenid mussels have increased dramatically over the past few decades across most of the Great Lakes, and Lake Huron is certainly no exception to that. That expansion brought with it significant changes to benthic communities, including substantial declines in important benthic species such as Diperia, which are key prey for many fishes, including lake whitefish. And as that expansion of mussels and decline of Diperia have occurred, of course, we've also seen declines in lake whitefish, as shown here in this figure of yield from the 1836 treaty waters. You'll note here that the declines are really primarily due to declines in lakes Michigan and Huron, which have declined by about 70% since the mid-90s, which was their peak. In contrast, harvest in Lake Superior, which doesn't have very many mussels in it, has remained relatively stable. So the overarching goal of this project is to determine whether we can improve lake whitefish production by removing Dreissenid mussels. Through engagement with lake technical experts, it was ultimately determined that the initial focus of this work should be on exploring whether removing mussels from spawning habitats can improve embryo survival. And while we're primarily focused on embryo survival, we're also collecting data on other life stages and processes, specifically on emerging sac fry and also on behavior of spawning adults. So this SWIM project, which we kind of view as a pilot mussel control project, is funded by the Great Lakes Restoration Initiative, but it's only one of the federally supported mussel initiatives. There's also what's called the Invasive Mussel Collaborative, which was established back in 2016, and their goal is to advance scientifically sound technology for invasive mussel control to produce measurable results. This Invasive Mussel Collaborative is currently in the midst of a structured decision-making process to develop a roadmap to inform research and management, a roadmap to inform site-level control of Dreissenids within the Great Lakes. So while this SWIM project is separate from the Invasive Mussel Collaborative, these two projects really feed into each other nicely. We work closely together, and they both feed toward our long-term goal of regional mussel control. So this SWIM project is organized into a handful of different teams, including a core federal team, a core planning team, and 5 implementation teams that cover various topics, including mussel control, which is led by Diane Waller, lake whitefish adult behavior, which is led by Tyler Funnel and Daryl Hondorp, lake whitefish embryos and larvae, which is led by Ben Tershak, and then formerly Ellen Marsden from UVM, and now myself. And then habitat mapping, which is led by Pete Esselman, and then nutrients, mussels, and algae, which is led by Marianne Evans. I want to take a quick moment to apologize to everybody involved in this project because I forgot to include a gigantic acknowledgement slide with all the people that work on this. But suffice to say, there's a lot of people that work on this project, and I want to quickly acknowledge and thank them for their efforts. I'll also point out the Great Lakes Commission provides backbone support for all of this work. And in total, we've got people from 14 different affiliations, including federal, state, tribal agencies, as well as academic institutions participating in this work. So this project includes study sites in Lakes Michigan and Huron. On the Lake Huron side, we've been conducting work in Thunder Bay on an experimental reef array, which I'll talk about in a few slides. On the Lake Michigan side, we initially focused on Good Harbor Reef, which is just northwest of Traverse City. But we've recently transitioned to focusing on the shoals in Big Bay to Knock because there are a lot more lake whitefish in Big Bay to Knock right now, and they're laying a lot more eggs, and that gives us a lot higher sample sizes and a lot better statistical power to detect any potential effects of mussel control efforts on lake whitefish egg deposition or embryo survival. So the design of this study includes measurements of many relevant variables, namely habitat mapping and estimates of goby and mussel densities, measurements of cladophora and benthic nutrients, analysis of lake whitefish spawning behavior through acoustic telemetry, and then measurements of lake whitefish egg deposition in the fall, and then embryo survival to hatch in the spring. So the time frame for this work includes 1 to 2 years of sampling prior to mussel control, intensive sampling during the year of mussel control to try to get at sort of immediate and near-term effects of the treatments, and then sampling for at least 2 years following mussel control to try to get at longer-term effects. And as I mentioned, we've got a few study sites for this project. In Good Harbor Reef, we just implemented mussel control last year in 2025. We plan to implement mussel control in 2026 in Thunder Bay and Lake Huron. And then in Big Bay to Nock, we plan to implement mussel control in 2027. So just a quick timeline here. Work for this project started back in 2022 when team members engaged managers and mussel experts and received their support. And then technical fisheries experts helped to define the scope and the sites for the work. In 2023, the experimental design was finalized and the sites were selected. And then the working groups that I mentioned previously were formed, and we received funding from the Great Lakes Restoration Initiative. 2024 was the first year of sampling. And then again in 2025 was the second year of sampling, and we also had that first mussel control effort implemented at Good Harbor Reef. And we identified Big Bay to Nock as a treatment site for Lake Michigan in 2027. Again, that's just because there are more lake whitefish there. And then this coming year, 2026, or I guess this year, 2026 is our third year of sampling. And we have mussel control planned for Thunder Bay in Lake Huron. So speaking of mussel control, this past summer, that mussel control team, as I mentioned, deployed 2 types of mussel control at Good Harbor Reef at a sort of hot spot of lake whitefish spawning activity. They deployed benthic mats, and then they also deployed benthic mats with carbon dioxide. They deployed 2 different types of this first treatment, the just benthic mat treatment. They had a polyvinyl mat and a canvas mat. Both of those mats were equally effective, but the polyvinyl mat was a little bit more durable and was reusable, unlike the canvas mat. I should say here that the idea with these mats is basically that they lay down on the bottom and they essentially suffocate the mussels. So the mussels die underneath the mats. The benthic mats were laid for 30 days, and we saw roughly 80% initial mortality of mussels underneath the mat. And then mussel densities remain low at 30 days post-treatment, likely due, at least in part, to predation of small mussels by gobies, which are kind of keeping them off of the rocks from recolonizing. The mussel control team and the nutrients, mussels, and algae team wanted to take some measurements at 60 days post-treatment as well for that benthic mat treatment, but they were unable to do that due to the government shutdown. And then they also deployed benthic mats with carbon dioxide. So these are mats that have hoses in them that allow for the pumping of water that's infused with carbon dioxide underneath the mat. And that helps to decrease the dissolved oxygen under the mat even faster, and it also lowers the pH, which can also be, and make it more acidic, which can also be lethal to the mussels. And so these treatments are much shorter. They're only about 4 to 5 days long. And we saw 98% initial mortality under those mats, which is really interesting. And then, again, the mussel densities remain low at 30 days, and in this case, they also were able to take a measurement at 60 days post-treatment as well. So the mussels did not seem to recolonize very effectively within that window. So to give you all a visual of what that control effort looks like, I have about a 3-minute video here for you all, and it should have started playing. Hopefully it'll tick over here in just a second. Yeah, we should be good. All right. Thanks very much to Diane's team and Marianne's team, or Diane's team and Marianne's team for putting that together, and for the Great Lakes Commission for also helping out with that. I have one more quick video here for you. This one's only about 15 seconds long, but it does a really good job, I think, of showing what that treated versus untreated areas, what they look like, kind of like what you saw at the end of that last video. So as the video starts here, the treated area is on the right and the untreated area is on the left. And you'll note that there's a really clear difference in the appearance of the mussels and the algae, and that the mussels in the treated area are nearly all dead, which you'll see as this diver kind of swims down toward the bottom. Okay, so that's what the mussel control efforts have looked like so far. As I mentioned, on the Lake Huron side, our study site's in Thunder Bay, which is near Alpena, and we're specifically focusing on a series of experimental reefs that Dr. Ellen Marsden and her team constructed back in 2010 and 2011. So these reefs feature 2 lines of 12 distinct rock piles each, and they're adjacent to 2 historically important spawning reefs that have been degraded by cement kiln dust, which is a byproduct of the cement manufacturing process from plants in the area there. These rock piles are really handy because they can be treated as experimental replicates, and some of them also feature pipes that are installed in the middle of the reefs that allow for monitoring water quality and other parameters at multiple depths within the reef itself, which gives us a sense of kind of what's going on down in between the rocks and not just what's going on top of the rocks. In addition to these experimental reef lines, we're also collecting data on what we call East Reef, which is right over here. And that's a semi-natural reef that's near these experimental reefs that we're treating as sort of an experimental control. So here's a diagram of our sampling across those 2 lines of experimental reefs. You'll note that half of the rock piles in each reef are 1.5 meters tall and the other half are 3 meters tall, as denoted by the blue and the yellow here respectively. So we're collecting fish eggs and larvae on 1 tall and 1 short reef in each of those 2 lines, as shown here by the stars. The red dots here indicate those monitoring wells, those pipes that I mentioned that are installed inside the reefs, and we're collecting water quality, nutrient, and other data on those reefs, as well as on other reefs in the array. So one of the nice things about these reefs is that they've been monitored as part of other projects for lake whitefish eggs in the past. So as I mentioned, the reefs were constructed back in 2010 and 2011, and lake whitefish egg densities from 2011 to about 2015 were roughly 240 eggs per meter squared on average. So sampling for this project started in 2024 and continued in 2025. There were some issues with the gear in 2024, so I wouldn't consider those estimates to be terribly reliable. But in 2025, we estimated that egg densities were about 61 eggs per meter squared on average. So this difference between 2011 to 2015 and 2025 could be due to, for example, lower numbers of lake whitefish spawning in the area or other factors. But there were some cases where egg densities were kind of similar between those periods. So for example, if you look at 2013 and 2025, they had relatively similar egg densities. Importantly, we didn't see any differences in the number of eggs per sampling bag across the experimental reefs or the semi-natural reef, East Reef, in 2025. So they don't seem to be favoring one location over another for egg deposition at this point. And it'll be interesting to see if there's any change in that pattern following the mussel control effort that we're going to be implementing here this summer. Another important note is that we did not capture any larvae, lake whitefish larvae, in our emergence traps in 2025. But we're going to be back out sampling actually starting tomorrow. I'll be on a boat first thing in the morning in Thunder Bay to check for emerging larvae on those reefs. There we go. On the adult behavior side, Tyler Funnell and his team have tagged 76 lake whitefish to date. And the telemetry data show that the whitefish spend most of their time on East Reef, right over here. But they also visit the experimental reef lines, which you can see kind of here and here. Within the experimental reefs themselves, there's somewhat similar usage by lake whitefish across the board, so they don't seem to prefer one particular rock pile over another at this point. And again, it'll be interesting to see if that pattern changes following mussel control implementation. Speaking of that, the mussel control team plans to implement these benthic mats with CO2 at Thunder Bay this summer. The mats look something like this. They've got that sort of polyvinyl mat, and then underneath they've got hoses that have holes in them that allow for that CO2 infused water to be pumped underneath the mat. We plan to treat 4 of the experimental reefs, and we'll be covering the entire reef with a mat in each case. Before, during, and after treatment, we'll be collecting data on water chemistry, benthos, and live mussel densities, both inside and outside of the treated areas. And finally, we're going to be sampling for eggs and larvae again this fall and moving in future years. and also in the spring to try to determine whether mussel control has had any effect on egg deposition or larval survival. And in terms of site selection for the control, it's going to be occurring on 2 of the reefs that we're sampling for eggs and larvae, shown by the stars, and two of the reefs that have the monitoring wells in them that we're measuring for many other variables. So I'm showing that here in the red shading. So in summary, we just completed our 2nd year of this project, which included mussel control on Good Harbor Reef with benthic mats. We saw high mortality of mussels and minimal recolonization within a couple months of treatment. Our primary goal here is to evaluate lake whitefish egg deposition and survival to hatch, but we're also measuring a lot of other variables, including mussel densities, goby densities, nutrients, and cladophora. The Great Lakes Restoration Initiative continues to support this project, recognizing state agency support. And we'll be controlling mussels in Thunder Bay in 2026 and Big Bay to Knock in 2027. And we hope to learn how mussels are affecting lake whitefish and how to continue to implement mussel control at larger scales. So if you'd like to learn more about this work, please check out the website that we have shown here, which includes some additional information, some other videos and animations. And with that, I'd be happy to take any questions.